Yeah, thank you so much. Thank you for your introduction. And it's my great pleasure to have a webinar with the Korean friends and to introduce our platform, no more single BCM based platform for molecular antibody discovery. And as a host introduced, my name is Yang Lu. I'm the CEO of Eurogene Biosystems. And the Eurogene Biosystems is a subtitle company of ABclonal. So today I will introduce, our single B cell based platforms for molecular antibody discovery. And this is what I'm going to talk about today. And first of all, I will give a brief introduction of our company, then to introduce the SMAP platform, then with some case studies. Eurogene Biosystems was founded at 2015 at Worcester, Massachusetts, and Worcester is second biggest city in Massachusetts and near Boston, and we only have like 40 minutes driving to Boston. And in 2016, Eurogene launched a single BCL-based molecular antibody technology platform to provide the antibody discovery services. And in 2018, Eurogene Biosystems established a subsidiary company in Wuhan, China, to expanding our CRO service to East Asian market. And at 2021, and Eurogene merged with ABclonal technology. ABclonal is a lead of reagents and service in global life science market. So Eurogene becomes a subtitle company of ABclonal technology. And in 2022, by the end of March, and Eurogene Biosystems have successfully delivered more than thousands of customer rapid molecular antibodies, and as well as other molecular antibodies to our customers. And you can see we list some of our lead customers in US market, include like Sanofi, Takeda, Moderna, Johnson & Johnson, Gilead, and MSD. This is, the introduction for our parent company, ABclonal. And ABclonal, we have like 3 sites, and one at Boston, and also as Eurogene Biosystems located. And the second is in our headquarters in Wuhan, China. And we have another research center in Shanghai, China. And we have the global marketing. We have several sales market in United States, and also we have some, sales office in Europe, including British, Belgium, and Germany. And we have the sales in Japan, and as well as in Korea. In Korea, and we have our subtitle company, ABclonal Korea, and to service our Korean clinic. So now I just would like to introduce a little bit about the technology background of our SMAP platform. And this is, the overview of our platform. And, first of all, and we will use multiple species like immunized rabbits, llama, and mouse, and as well as a human. Human generally, you cannot immunize the human being. So we will use some, infection disease recovered human and collect their samples, for example, like the HIV infected, or currently like COVID-19 infected. I will show you a case study later on. And then after immunization, or collect the PBMC samples from human, and we will get the either spirochetes or PBMC from the animal and the human. And we will prepare the spirochetes, and then the third step, we are going to sorting, and we will use antigen-specific sorting. We'll try to use antigen to enrich the B cells, and combined with the FACS sorting. And after that, that was proprietary technology of our company. We can culture the B cells. Because the B cells are primary cells, generally it's very difficult to culture, but we can, culture it and expand it for like 2 or 3 weeks. And after that period, and in the supernatant, they have some, like superintendent, they have some antibody, secret antibody in there. It's allow us to do some, high throughput screen like ELISA, FACS, as well as the Western blot and others to identify the positive clones. Then we, from the positive clones, we will clone out the, variable region of the antibody genes from the positive clones. Then try to in vitro recombinant expression them, and try to validate and screen again. By the end, then we will final validation and scale up to express antibody use our in vitro systems in mammalian cells. That was of our technology that was different to the traditional hybridoma technologies, and we directly sought out the B cells secreting the antibodies from the animals or human. Yeah, currently majority we focused on rapid molecular antibody development with this technology. So this is typical, rapid molecular antibody development schedules. We divide it to the 3 phases. Like the phase 1 is immunization, and the phase 2 is like a B cell sorting and cloning. Phase 3 is a final validation and scale up expressions. So then we will final delivery is a sequence of all discovery antibodies. So it's not like hybridoma cells. We don't deliver the cell lines to customer. We just deliver a sequence, and based on sequence or the plasmid we deliver, you can do recombinant expression by yourself, and it's easy and it's safety. You don't need to worry about when you store your cells, when the liquidation tanks is gone and your cells is died and everything. There's nothing to worry as long as you have the sequence information there. And we can also minimum delivery 100 microgram of the antibody protein A purified recombinant rapid antibodies to our customers. And then the typical, the turnaround time is 10 to 17 weeks. It depends on how long you need to immunize animals to get the high enough titers. It's definitely depend on your antigen itself. But we also have fast tracks, and we shorten our phase 2, shorten time from here like 2 to 3 weeks to 2 weeks. Then we can even deliver antibody fast, and the total turnaround time will 11 to 15 weeks. So this is a comparison for the currently 3 major platform for antibody development. The traditional, and I think most people know that, it's hybridoma technologies, and another is display, include the phage display and yeast display, then the single B cell sorting and cloning technologies. You can see, our technology is significantly, save time compared to hybridoma and phage display, and after the immunization, because immunization is the scale timeline is the same for all technologies, but after that one, our technology is fast and the efficiency is higher, and we can get the nature pair of the antibody. And also we deliver plasmid, as I said, carrying the antibody sequences. And the final product is more stable. And we know the antibody sequence, and we deliver the sequence to customer. The antibody diversity is higher. And also we can do most host species, including rabbit, monkey, human, and also llama and mouse. Yeah, and ABclonal and the European biosystems, not only we focus on our SMAP platform, we also have different immunization platforms to make our platform is more than other companies. We can do traditional peptide and protein immunization. We also can do cell line and with DNA immunization, and also the VLP immunization and mRNA, most recently the mRNA immunization. For that one, we cooperate with Moderna and the famous company to develop the COVID-19 mRNA vaccine. So we also can do the antibody engineering, include the antibody fragment. We can generate the different form of the antibodies. and we can do affinity maturation, and also for the isotope selection and swapping, we can swap the the isotope antibodies. And for the therapeutical purposes, we can do the antibody humanization with AI assistant platform, and also for the FC engineering. Yeah, this is majority is for the therapeutic antibody development, and we can analyze the antibody sequences and can do the in silico developability screen to de-risk the final development at the beginning. When we know the antibody sequences, we can do the analysis with the AI, artificial intelligent at the platform assistant, and to do all kind of analysis and to evaluate the developability of the leading compounds. Then we will deliver that to our customer and to help them choose the best antibody to develop the therapeutic antibodies. Yeah, this is just a few example how we do the developability scanning to de-risk. Yeah, like give examples, we can compare the different oxidations, like those levels. Then we pick up the best one and go to the humanization, because based on the function assay, you can see all the four clones is similar. It's very difficult to make a decision and which one is the best and which one have less risk to develop the therapeutic antibodies. But by this analysis, it can help you try to de-risk. Yeah, this is we talk about our platform, how to help people to select antibodies, to develop the therapeutic antibodies. And at the beginning, I said, for our platform currently, we majority focused on develop the rapid molecular antibodies for our customers. So why we choose rapid molecular antibodies? They have some reasons on there. First of all, the rabbit have the unique immune systems. They have two sides of the unique immune systems. One is as we know, for the small antigens like those heptans, peptides, it's not the antigen present is not via MHC molecules on the T cells. It's via CD1 antigen presentation pathway. But in rabbits, they have more advanced CD1 systems compared to the mouse and the human. So it make the rabbits is ideal for like those small antigens presentations. So it is good for the PTMs, post-translational modification, specific antibody development. And also it's good for small chemical compounds antibody development, as well as anti-nucleotides or polysaccharides, and as well as traditional peptides. It's easy in rabbits to rise the good titer with peptides compared to in mouse. And the second is the rabbit have better developed immune systems. As we know, in human and in mouse, their primary cultivar development is stopped after birth. But in rabbits, even after birth, their primary, B cells repertoire is still, developing for like 2 or 3 months. It can increase the diversity of the antibody developed. So in other words, in human and mouse, after birth, when you inject antigens to human and mouse, and they just use the somatic mutation to help the antibody maturation and the development. But in the rabbits, except the somatic maturation, it still can happen like a gene conversion, like that. So it can increase the diversity of the antibodies. Also, the rabbits have gut associated lymph node tissue, and that was very unique. It doesn't have in any other species. It can help the rabbit to develop the higher affinity antibodies. So then also the unique part for the rapid antibodies, in the human, for human and mouse, for other spatial antibodies, generally people know the heavy chains contributes more to the antibody antigen binding. But in the rabbits, people find that the light chain is also contributed to the antibody and antigen binding. And the light chain, the length is more distributed than mouse and human. Is also contributed to the diversity, great diversity, and a higher affinity of the rapid molecular antibodies. So this is application for the rapid molecular antibodies in science and in industry. And first of all, the people use it as two antibodies for academic researches. It's good for the habitat, as I said before, or higher conserved epitopes, or also for the post-translational modifications like phosphorylations, methylations, and others. And also the rapid molecular antibody is good for the in vitro diagnostic reagents. It's because its highest affinity is caused the rapid molecular antibodies have the higher sensitivities. So it can help to develop a higher sensitivity in vitro diagnostic assays. Right now the rapid molecular antibodies is used for the therapeutic antibody development. There's two FDA approved, antibody drugs is original from the rapid molecular antibodies. One is VEGFR, is developed by Novartis, is used for the eye diseases, for treatment for eye diseases, and another is for CGRP, is just FDA developed, approved. They have another several of target is in the phase 2 or phase 3 clinical trials. Because of the great diversity of the rapid molecular antibodies, is make it ideal for the anti-EG type antibody development. The anti-EG type antibodies is very important when you develop the therapeutic antibody drugs, when you try to apply it to the clinical trials. It can be used for the PK studies and for the immunogenicity analysis, and as well as for the vaccine development. So this is an advantage of our SMAP platforms, and we call it universal, stable, efficient, and rapid. And universal, it means it not requires hybridoma fusions, so it can develop the molecular antibody from multiple hosts, as I said before, and also stable because it develops validated sequences and also use the recombinant expressions. And the important thing is for the industries, we don't require any loyalty or an extra license fees for our customers. In other words, when we deliver the antibody sequence to you, and you own all the RPs, you can manufacture it and do engineering by yourself. Is no any limitations, and the efficiency then it includes assured monoclonalities and also the excellent diversity with efficient functional assays combined. And the repeat is we can develop antibody in short turnaround times. So in the next few of slides, I will present some case studies and for our audience. The first studies, this project is, started from the 2020s with the onset of, the COVID-19, and the lucky and the unlucky is our company's, headquarters in Wuhan. And, I think you might know the COVID-19, and the first is started from Wuhan, China. So we have the information early, and we initiated the project. We try to use combined, our single B cell platforms with rabbits and also with our, proprietary DNA immunization systems to develop the, we try to develop the broad neutralization antibodies against the COVID-19. And, we use those 3 DNA immunization followed by 2 boosts and a final boost with sub-Q. Then we collect the spleens and we do the B-cell sortings. You can see, our ELISA positive ratio is very high, and we only have like 1152 single B-cell clones, and we get 350 clones ELISA binding to the S1. Then among that, we find 114 clones binding to RBD domains, and the 22 clones and have some neutralization activities, and 23 blocks to the ACE2 and RBD interactions. This is some of the raw data, and by the end we find out 1 clones, the clone number is 1H1, have some great neutralization potential. It can bind to all current Omicron variants. And this is we do the the virus neutralization assays. They can show the broader neutralizations. And then we collaborate with Nanjing University of China. This is a part of unpublished data. And the previous works we already published, at least here, published it in emerging microbial infections in 2021. And then, we just focused on this clone because this clone shows a broader neutralization activities. We use the CRY-EM, try to learn about its crystal structures. And the interesting thing is we find, these clones is not competed RBD binding with ACE2, that represent the class 3 antibodies, which bind to both up and down of the RBDs, the outside of the ACE2 receptor, binding motifs. And this data we just submitted for another publications. Yeah, and I think this part is show high specific antibodies, rapid molecular antibodies. It can differentiate the wild type and the mutant, and we can distinguish the only 3 amino acid difference, the 2 proteins, wild type and mutants. The antibody we developed can recognize the like the mutant antibodies, mutant proteins, and without binding to the wild type proteins. And also, this shows a greater specificity. Also, shows a rapid is a good model for the small chemical compounds. We developed a panel of rapid molecular antibodies, which can recognize a single nucleotide like adenine and cysteine and G and T. It can recognize like DA, but no binding to any other like nucleotides. And these antibodies we developed for the BGI, the second generation sequence company of China. We help them to develop the second, actually third generation of the NGS kits. So then I will talk about, like the rapid molecular antibodies for the PK, PD, and ADA antibody development. And this is like just a global, introduction of when you need the PK, PD, or ADA antibodies. Usually, those antibodies is needed to required to the preclinical and all the clinical in phase 1, phase 2, phase 3. It will help you to study your antibody drugs metabolism on the human bloods. So generally, when your clinical studies started, you need to develop the antibodies, antibody reagents for these purposes. Because they have 3 different type of the antibodies for the PK studies. Type 1 is a blocking antibodies. It can block your antibody drugs binding to your the antibody drugs targets, the protein targets in your human bodies. And the second one is no blocking. It will not infect your antibody drugs binding to its targets. And the third one is specific, only specific to the the antibody and your target's complex. It's very difficult to develop for the type 3 more antibodies. So then people generally develop the type 1 and type 2 antibodies, then to do some mathematics calculations to find out they have how many your antibodies still binding to your antigens. And this is one of the case studies. It also shows higher positive ratios. We can get about we thought about 2,000 single B cell clones and get about 20% of the positive positive clones. And among that one, we can quickly do like blocking assays to distinguish how many blocking clones and how many no blocking clones. So this is show the case study show development of the PTM, post-translational specific antibodies. For this kind of antibody is very interested to the academia users. People will need that as a tool to discover their the protein's phosphorylation status in cells. So we have developed a lot of these kind of antibodies with different colleges, and they publish a lot of papers in very good journals with our help. And you can see for these case studies, we developed antibodies recognize the phosphorylation site of this protein called PTEN at Y220s is very specific. You can see we use these antibodies for the IgG standings, then show specific standing with the phosphorylation proteins. So these studies show you, how we develop the therapeutic antibodies, for the customers. We have done lots of those projects, but for the legal issues is confidential. We cannot disclose the antibody this information to other clinics without permission, so we cannot say anymore. So then we decided to develop the one target like the CD40s by ourselves, just to show how this platform can help to develop therapeutic antibodies. So the good thing is we can combine the in vitro report assays very early stage. You don't need to go to the by the end to validate the antibodies with the function assay. We can do it very early, even with a single B cells supernatant. You can see, we sort out 1152 single B cell clones. Among that one, we find 53 cell surface binding clones. That means FACS positives. But the FACS positives not means these antibodies have some function, and we can do further, in vitro report assays to narrow down 16 functional clones. They have some unlist functions. Then we just focus to study these 16 clones, then try to clone them out to study its affinity, then to if it's good, then to do the humanization. Yeah, then another part I said, because of the high specific of the rapid molecular antibodies, make it ideal for the IgG immune histological standings. It's a gold standard for the IgG standings, and more and more people use it as a gold standard for the companion test, like for the drug candidates like the PD-L1 and also the VEGFs. And this is the antibodies we developed. They can show the very specific standing in the patient tissues and no any background standing in the normal control tissues. And also, we can use previously in all the case studies, we show you how we can develop the rapid monoclonal antibodies use the SMAP platforms, but we also can, develop the llama nanobodies, use SMAP platforms. This is one of the studies. We try to develop the llama VHH antibodies and used for AAV delivery. And actually, we have our own farms in China, Wuhan, and we have a collaborative farms in Boston, United States. They have both rabbits and llamas, and we can help to do the llama immunization and also with the single B cell sortings to discover the llama VHH antibodies. So this is another case is show how we develop the normal biomarkers from the patients, and this is we cooperate with another hospital in China called Tongqi Hospital. Is almost top 10 hospital in China, and then because by the clinical studies they find out some molecules is associated with like some heart diseases. So they try to develop the antibody pairs can use for the early diagnostic to detect those protein levels in blood to early development of the diseases. Then they collaborate with us, and we help them to find out the paired rapid monoclonal antibodies used for sandwich ELISA and as well as for any other ACEs. And they used our antibody pairs, to develop the diagnostic kits to analyze the patient samples. They can show the biomarkers they are interested, show the significantly higher in the patient compared to the controls. So by the end, I know we just show you, our platforms, and we just focused on SMAP platforms for the antibody screening, and we provide one-stop antibody development services from the antigen design, antibody screen, to antibody engineering and antibody production. For antigen design, and we can help you to design the antigens, either use peptide or use proteins. If the proteins, we can help to do the recombinant protein expressions, and we also can do the cell immunizations, VLP immunization, as I said before. And also, for antibody screening, we focused on our SMAP platforms. We do multiple species antibodies development, and also, as I said before, we can do antibody engineering as well as antibody production. This is our instruments currently we have, and we have 3 FACS sorting instruments to do the single B cell sort for the customers, and we will have this machine is very similar to the octets. They call it ABclonal. They can help us to measure the affinity of the antibody and the antigen binding, and we have 3 of them. And this is ABclonal. ABclonal 3000 is also measure the antibody and antigen binding. It's more accurate. This one is for the high throughput. This is effects analysis instrument to help you to do effects analysis, and we have multiple HPLC instrument as well. So thank you so much for attending these talks. So yeah, if you have any question, just let me know. Thank you.